Hey there!
Hey.
I'm Allie Astrocyte, and you're tuned in to Neuro Transmissions. In our last few episodes, we talked about resting potentials and action potentials, the language used by neurons to communicate their signals from one end of the cell to the other. But how does that message get passed from one neuron to the next?
Yeah.
Each neuron is separated from its neighbors. How do they do it?
Yes.
Now I get to talk about my favorite part of the neuron, the synapse. Before we go on, if you haven't already, you may want to brush up on the anatomy of the neuron with this video. And you might also want to watch the videos about resting and action potentials. As we discussed last week, your neurons use ions and electrical potential differences to transmit signals from one end to the other. As that signal reaches the end of the axon, it comes to a structure called the synaptic terminal. This structure has several parts. The side of the cell sending the signal is called the presynaptic terminal. The gap in the middle is called the synaptic cleft. And finally, on the other cell receiving the signal is the postsynaptic terminal. There are two basic categories of synapses: the electrical synapse, or gap junction, and the chemical synapse. The electrical synapse is the more simple of the two, allowing direct ionic current transfer between cells to pass the action potential across the synaptic cleft. These types of synapses have very little space between the pre- and postsynaptic terminals, about 3 nanometers, and use specialized proteins called connexins to form channels that allow the ionic current to pass between cells. It's sort of like a skywalk between two buildings. They are still two distinct buildings, but it's easy to send a message across to the next building because you can just walk across the bridge. Because the gap is so small, electrical synapses allow cells to respond very quickly to signals, and mean that the cells connected tend to react together. Because of this, they are important for tasks that require very fast signaling, like defensive reflexes in animals. The other type of synapse, the chemical synapse, is wider and more complex, with slower signaling. In these synapses, the synaptic cleft can be 20 to 50 nanometers wide.
Whoa.
Still pretty tiny, but a lot bigger than a gap junction. Because of this, using ions to directly signal onto the next cell isn't practical. There is no sky bridges connecting these two buildings. Instead, chemical synapses came up with a different way to send their messages. Remember the presynaptic terminal? Let's think of it as a cellular post office. In the presynaptic terminal, you can find these itty-bitty spherical structures called vesicles. Think of them like envelopes. These vesicles are little pockets of membrane that hold the information to be transmitted, keeping the message contained until the post office of the synapse is told to release the information. Inside each vesicle are chemicals known as neurotransmitters. Quite literally, these molecules transmit information between neurons. These are like the letters contained within the envelope. As the signal arrives into the presynaptic terminal, it tells these vesicles to move to the edge of the cell, where the vesicle membrane fuses with the cell membrane and opens up, dumping its contents into the synaptic cleft. Once the neurotransmitters reach the postsynaptic terminal, they attach to receptors. You can think of these receptors like a mailbox. The neurotransmitter binds to the portion of the receptor that sticks out of the cell membrane, and the information they contain is interpreted by the receptor into some kind of response. Sometimes that response means opening an ion channel attached to the receptor itself, letting ions flow and changing the electrical potential of the cell, like we talked about in our last video. Often, the receptor passes the message along to other proteins that rush off into the cell to make other kinds of changes that will affect the electrical potential. There are many different kinds of neurotransmitters, but 2 of the most common are glutamate, found at glutamatergic synapses, and GABA-aminobutyric acid, or GABA, found at GABAergic synapses. These 2 neurotransmitters usually have opposite effects on the cells they are signaling. Glutamate is considered excitatory, while GABA is considered inhibitory. Let's talk about each of these separately. There are 2 main types of glutamate receptors: the N-methyl-D-aspartate receptor, NMDAR for short, and the alpha-amino 3-hydroxy-5-methyl-4-isoxazolic propionic acid receptor, or AMPAR for short. Thank goodness.
Whoa.
When glutamate binds to an NMDAR or an AMPAR, it opens a channel in the cell membrane attached to the receptor, letting positively charged ions move in and out of the cell. Because these receptors are part of an ion channel, the binding of glutamate results in a very fast response, which lets the cell rapidly depolarize and start transmitting the action potential down to the other end. This is why they are called excitatory synapses, because glutamate binding to the receptor excites the cell to spark an action potential, making the postsynaptic cell spike. Inhibitory synapses have the opposite effect. The GABA binding to the receptor actually represses signaling, blocking the postsynaptic cell from firing. GABA can bind to two kinds of receptors. The GABA-A receptor is much like the AMPARs and NMDARs, opening a pore that lets negative ions into the cell, which makes it harder for the cell to fire an action potential because the inside of the cell becomes even more negative than normal. GABA-B receptors are called metabotropic receptors. They're the kind I mentioned before, where the receptor sends off a messenger protein to signal elsewhere in the cell, generally resulting in the same thing, making the inside of the cell more negative to prevent it from firing a signal. Hang on. If your brain has to fire action potentials in order to send a signal, why would an inhibitory signal be useful?
Yeah.
Well, it can help prevent your brain from sending too many signals at once. Too much signaling can be just as bad as too little. It helps your cells fine-tune their firing. For example, a cell might receive an excitatory signal followed quickly by an inhibitory signal, so it turns off very quickly and can't fire again right away. So now you have a broad overview of the synapse. It's crazy to think that this structure gives your brain the precise tuning and communication it needs to let you walk, talk, breathe, eat, sleep, think, and be who you are while moving through the environment around you. But it's true, our brains wouldn't be much good without the synapse. Thanks for watching. If you like this video, give it a thumbs up and subscribe to become a Brainiac. Tune in next week as we talk about all of the different major structures of the brain in our episode on neuroanatomy. Until our next transmission, I'm Allie Astrocyte. Over and out.